Hello, dear students. Welcome to Margdarshan Series. So, in this video, we basically talk about animal tissue culture. So, first thing is what is tissue culture? So, culturing of any cell or tissue in a culture medium or outside a cell is called as basically tissue culture. So, cells of an animals exist as a organized tissue matrix which required for the control growth and differentiation. So this cell from the intact organism, for suppose this is an intact organism, for suppose this is a mouse. Okay, from mouse we isolate its liver, and from the liver we culture its hepatocyte. So hepatocyte culture in culture medium. So that is basically called as a tissue culture. So the cell from the intact organism may be isolated, maintained, and grown in a in vitro culture media aseptically. So what is aseptically? Aseptically means free from microbes. Free from microbes, suitable for the mixture of nutrients and growth factor. This process is basically called as animal tissue culture. What this is called? This is called as animal tissue culture. So we will talk about primary culture. What is primary culture? So primary culture is prepared by inoculating the cells, inoculating cell directly from an organism into a culture medium, as told you here. So this is basically called as primary culture. Directly we take a cells and culture in a culture medium. So that is basically called as primary cell culture. Next is this culture has a limited lifespan. So this culture have limited lifespans because except cancer cell. So except cancer cell, the other cells having limited lifespan because after certain division it will go for senescence. Okay. Next is the limited proliferation capacity basically reflects the limited proliferation capacity reflects the progressive shortening of the cell telomers. So after certain differentiation, so cells telomers started shortening. This respective DNA sequence associated protein that can cap the end of each chromosome. So there are some proteins that is basically uh, get accumulated at the end of the each chromosome, which prevents them further division. Okay, so there are two major type of primary cell culture. So primary cell culture having two major type. One is adherent culture or attachment culture, or sometimes it's called as adherent culture, and suspension culture or non-adherent culture. Suspension culture is also known as non-adherent culture. So adherent is also known as anchorage dependent culture. Adherent is also known as anchorage dependent culture. So for the survival of cell, it needs to attach to the surface. Without it cannot survive. So for in case of anchorage dependent or adherent cell culture type, the cell has to be attached to the cell has to be attached to the surface of the culture medium for the proper growth and development. Without it, it cannot survive. Example of your attachment culture is your kidney cells. So kidney cells are type of your adherent culture. This there from here question can be asked. Okay, example question can be asked. Next is suspension culture. So suspension culture is also known as non-adherent culture. Sometimes it is also known as floating floating culture. Sometimes it is called as floating culture. Floating culture, non-adherent culture, or this is basically anchorage independent cell. This is basically anchorage independent cell. So basically it does not require any attachment to the cell surface. So it does not require to attachment to the cell surface. So it can be float on the culture medium for growth and development. Cells will multiply when they are suspended into the cell culture medium. So cells will multiply. So when they undergo when the cells are basically in a suspended condition, so they basically that is the ideal condition. This floating condition is basically ideal condition for suspension culture cell. So example of this is lymphocyte culture. So blood lymphocyte culture is a typical example of suspension culture. You can see in case of adherent culture, the cells are completely attached to the base of the membrane. But in case of suspension culture, cells are basically floating on the culture medium. Okay. Next is cell line. So what is cell line? When primary cell culture undergoes subculture. So we get primary culture. So primary culture, how we get? We when we directly inoculate the organs into a culture medium, we get primary culture. So from the primary culture, when we subculture it, subculture means from one cell we take and this cell will again grow in a culture medium. So we get secondary cell culture or also known as cell lines. So cell line means what? When primary cell culture is subcultured, we will obtain a cell lines. So subculture means it refers to the transfer of cells from one culture vessels to another culture. So the cell lines are basically divided into two major class. So one is finite cell line, another one is infinite cell line. So two major type of cell lines are there. One is finite cell line, another one is your infinite cell line. So what is finite cell line? So finite cell lines means that will undergo cells will undergo only a finite number of division in a culture and eventually undergo senescence. So this finite cell line basically having a limited number of division in a culture medium. So after a limited division, so this finite cell line undergoes senescence or it will start a dying. Next is it has a possible number of subculture or passages. It has a possible number of subculture or passages. Normal mammalian cells generally have a finite number of life cycles. So normal mammalian cells, so these are basically having finite number of life cycles. So after 5 to 10 or 10 to 15 division, it will start senescence. After finite number of divisions, cells stop dividing. So these type of cell basically exhibit property like contact inhibition. So this type of cell basically exhibit property like contact inhibition, density limitation, and anchorage dependent. Okay, three major that can be exhibited by finite cell line is contact inhibition. So when the concentration increases, the cells started contacting with each other. They started the inhibition process started. Density limitation, when the density of the cell culture increases, that will lead to a cell death. And next one is anchorage dependent. Next is some of the cell lines basically avoid senescence. Some of the cell line basically avoid the senescence and give rise to a continuous cell line. Some of the cell lines basically avoid senescence and give rise to a continuous cell line. A cell line that has the potential to be subculture indefinitely time. Indefinitely. That means indefinitely period of time you can subculture it. So this type of cell line basically called as infinite cell line. So infinite cell lines are also called as transformed cell line. Why transformed? So there is a some of the transformation occurs in this type of cell line so that it will be subcultured for a infinite period of time. So due to alter growth property of a immortal cell. So basically example is basically your cancer cells. So cancer cells are typical example of infinite cell lines. It may occur spontaneously or induced by carcinogens or virus. So these changes from finite cell line to infinite cell lines. So this can be occur spontaneously or induced by some of the carcinogens or viruses. Transport cell line does not exhibit the property of contact inhibition. So they does not have the property of contact inhibition, density dependent inhibition, or proliferation and anchorage dependent. These are the few examples of property that can be exhibited by infinite cell line. So you have to remember what are the property exhibited by the finite cell line, what are the property that is exhibited by infinite cell line. Next is transport cell line. So transport cell line has an abnormal chromosome number and overproduction of different proteins. So this transfer cell line having abnormal chromosome number because there are abnormal in cell division. That's why the chromosome number will also abnormal followed by overproduction of different proteins. So there will be overproduction of different proteins. Next is what are the characteristics? So a transformed cell having what are the following characteristics that is basically exhibited by transport cell line. So first in genetic abnormality, the normal genetic sequence which we will get in our finite cell lines, we will not find any uh, genetic similarity in case of infinite cell line. So there is the genetic abnormalities is there. Next, in chromosome abnormality, the chromosome number is basic chromosomal abnormality is also seen. Loss of contact inhibition. So as you have seen that in case of finite cell line, when the cell started contacting with each other, there is a inhibition will start. But in case of transformed cell line, no contacts of inhibition property is there. Next is encourage independent growth. Encourage independent means there will be a growth where they can grow like a floating cell. Okay, next is reduced growth factor requirement. Now the growth factor requirement will be reduced. There will be a biochemical changes, increase glucose transport, increase lactate production. So these are the few characteristics exhibited by infinite cell lines, or called as transform cell lines. Infinite cell lines or transport cell line. Next example. So example of cell lines is 𝘂𝐻𝑘. 𝘂𝐻𝑘 is Syrian baby hamster kidney. These are the few cell lines that is basically used in different biomedical research. So 𝘂𝐻𝑎 is baby hamster kidney, Syrian baby hamster kidney. CHO stands for Chinese hamster ovary. Next is HeLa. HeLa is a human cervical carcinoma cell line. So this was derived from a 31-year-old woman named as Henrietta Lacks, so who died due to cervical cancer in 1951. So this HeLa cell line, so basically isolated from a person named Henrietta Lacks, that who is died in 1951 due to cervical cancer. So from her body, so this type cell line was isolated. Next is L. So L is basically mouse connective tissue, or also known as mouse fibroblast. So this is the first cell line. This is very, very important from exam point of view. Which cell line is the first cell line that is basically isolated, that basically developed? So remember L, that is mouse connective tissue or mouse fibroblast. This is the first cell line that is obtained from the mouse subcutaneous connective tissue. The species is mouse. Mouse subcutaneous connective tissue by exposing the culture cell to a chemical carcinogen. Next is MRC5. MRC5 is a human embryonic lung. So MRC5 is human embryonic lungs. Vero is African green monkey cell line. WIS is human amnion cell line. MPC11 is mouse myeloma. So these are the basically the cell lines that basically used in different biomedical research. Okay, next we will talk about the growth cycle. So there are cell lines. So these cell lines are typically exhibit different growth phase. Growth phase. These are like lag phase, log phase, plateau phase, or after that another is there, and decline phase. So log phase is so first is lag. So lag phase is the period where zero growth when so the here the period of zero growth. So question can be asked, which of the following phase is a period of zero growth phase? So lag phase is a period of zero growth phase where the cells are first inoculated into the growth of the growth medium. So initially what we have to do is, so to the culture medium, we initially inoculated this cell into the culture medium. So cell first what it will do? It will start acquainting with this culture medium, start surviving itself in the surviving in the culture medium. So here the period of growth is basically zero. The period of growth is basically zero, and the first it is inoculated in a growth medium. The length of this phase basically depend upon the type of cell and their metabolic state during inoculation. So the length of this phase. So how at what till what duration this lag phase will continue? The length of this phase basically depend upon the types of cell. What type of cell you are basically inoculating, and the metabolic state during inoculation. So what is the metabolic state of cell during that inoculation? Next is during this period, cell recover from trypsinization. So during this period of time, lag phase, cell basically recover from trypsinization, reconstruct their cell skeleton, and secrete matrix to aid the attachment and spread out the substrate. So what is trypsinization? So So if this is a culture medium, so trypsin basically added. So trypsin basically added for the for this trypsin uh, basically help the cell to detach from the base of the culture medium. Okay, so that is basically called as trypsinization. So they reconstruct their cytoskeleton. So these cells try to reconstruct their cytoskeleton, secret matrix. Okay, secret matrix to aid the attachment and spread out on the substrate. Next is log phase. So log phase is also called as exponential growth phase. Is a period of continuous cell doubling. So first, what happens? So cell introduced in the culture medium, they recover from the trypsinization, try to reconstruct their cytoskeleton, secret matrix to aid the attachment spread out on the substrate. So after that, now the cell will try to divide itself. So this is basically called as exponential growth phase. This is a period of continuous cell doubling. Within this period, basically continuous cell doubling takes place. So animal cell normally exhibit a doubling time between 15 to 25 hours. So between the time of 15 to 25 hours, so animal cell started doubling. So this time required is 15 to 25 hours. Next, the length of the log phase. So length of the log phase basically depend upon the seeding density, the growth rate of the cell, and the density at which cell proliferation is inhibited by this density. So basically, what basically log phase depends. So log phase, the length of the log phase. So at what time period the log phase will work. So that basically depend upon the seeding density, the what is the concentration of the media, the growth rate of the cell, what is the cell's growth rate, and the density at which cell proliferation is inhibited. Okay, so that these things has to be that is the these things are the key factor for the determining the length of the log phase. Next is plateau phase. So plateau phase is the stationary. This is also known as stationary phase. This is a period after growth when the no change in the culture cell density. You are not adding any further media or any culture, any growth sources to this culture medium. This phase occurs when the nutrient have been depleted or inhibitory metabolites are accumulated in the cell. When the nutrient which you have added for the cell has been completely, we can say, deteriorated, and the cell after the cell metabolism is, after metabolism there will be accumulation of metabolites. So cell, so that basically cell after the metabolism they started accumulating the toxic metabolites into the culture medium. So when the concentration of the toxic metabolite increases, that will leads to death of the culture medium. All the available growth surface is occupied. All the available growth surface is basically occupied, and all the cells are in contact with the surrounding cell. Further growth can be obtained by adding fresh media. So uh, if you want to reculture this culture medium, so you have to add the fresh culture medium to this culture. So you can see first phase is log phase. This is basically initial introduction, initial introduction of cell. So after that, log phase. Log phase is the basically exponential growth phase. Exponential growth phase. Next is stationary phase. Stationary phase is where the growth and death is equal. When the growth and death is at equal rate, so we call it as a plateau phase, or sometimes it is also called as stationary J phase. Next one is your death phase, where total when the culture medium will started, you can see the green line going downward. That basically indicating the culture medium started. The cells have started dying. Okay. I hope everyone is clear up to this. So next is, so we talked about cell, we talked about their growth medium. Next is where the cell will grow. So the cell will basically grow in a culture medium. So let's talk about culture medium. So this culture media, this is basically a typically this is a growth medium. This culture medium is nothing but typically a growth medium that is basically composed of a amino acid. What are the composition of? Remember, so the this growth medium basically composed of amino acid, vitamins, inorganic salts, glucose, serum as a source of growth factor, hormones, and attachment factor. So these are basically example of the component of a culture medium. So first is your amino acid, vitamins, inorganic salts, glucose, serum, hormones, and attachment factor. There are two major type of culture media that is used for the animal cell culture. First one is natural media. Second one is your synthetic media. Two type of media basically used for the culture medium. One is natural media, another one is synthetic media. So natural media basically consisting of naturally occurring biological fluid and used for the wide range of animal culture. So these basically natural occurring culture media basically occurring biological this basically naturally it is consisting of naturally occurring biological fluids. So biological fluid are used as a culture medium and it is used for the wide range of animal cell culture. So this is basically used for the wide range of animal cell culture. The major disadvantages of natural media. So what is the major disadvantages? Poor reproducibility. So this major disadvantages of this natural media is poor reproducibility. Okay, due to lack of knowledge of exact compensation of the natural media here. So there is a lack of knowledge about exact composition of the natural media. So the exact composition of the natural media is not known. Next is artificial media. So what is artificial media? So artificial media basically contains partly or fully defined components that are prepared by artificially by adding several nutrients such as organic and inorganic nutrients. So artificial media are supplied with serum and suitable organic components which support the survival of cells in culture. Okay, so these are basically about detail about the culture media which are basically used for different purposes. Let's talk about a culture media contains words. So culture media having different properties. So culture media has to be maintain the physical property. So what are the physical property it has to be maintained? First is pH. So pH of the culture medium should be at optimum pH, that is around, so you have to remember the value 7 to 7.4. So at this point, so the growth is maximum. Growth is maximum. Next is second point is carbon dioxide, a bio or bicarbonate and buffering system. So carbon dioxide, bicarbonate and buffering system. This is also required for the proper growth and development. So carbon dioxide concentration is 5% use. 5% carbon dioxide concentration is basically used. Okay. Next is oxygen. So oxygen supply for the aerobic respiration. Next is temperature. So temperature should be at a optimal rate. So that is around 37 degrees. So temperature is around 37 degrees. It should be optimal. Next is osmolarity. The osmolarity of human plasma is around 290 milliosmolarity per kg. So it has to be maintained for the proper growth and development. Next is balanced salt solution. So phenol red is serves as a pH indicator. So here phenol red is serves as a pH indicator for the pH change detection. Next is serum. Serum is this is a biological sample. So this is serum is nothing but this is a biological biological sample. So this is basically used to basically serum, scalp serum, fetal mobile serum, horse serum or human serum can be used for the growth and development. So this is basically growth factor for promoting this. Remember, this serum is basically act as a growth factor for promoting cell proliferation. So this is basically act as a growth factor for promoting proliferation of the cell. So other growth factor that is required, these are basically platelet derived growth factor, fibroblast growth factor, epidermal growth factor, vascular endothelial growth factor, and insulin like growth factor, basically IGF-1 and IGF-2. Okay, so these are basically the composition of a culture medium. Okay. So the whatever we have discussed is these are basically the composition. Sometimes question is asked about these amino acids, vitamins and glucose. So glucose is basically behaves as a carbon source. Okay, glucose is act as a carbon source. Okay, so glucose is basically act as a carbon source. Next, next is artificial media. So what are the different type of artificial media used? So four different type of artificial media is used for the growth and differentiation. First one is serum containing media. So serum containing media example is your fetal mobile serum. Serum containing media is a fetal mobile serum, that is the most common supplement for the animal cell culture media. This is the most common supplement. Fetal mobile serum is the most common supplement for the animal cell culture media. So this fetal serum containing media basically contains amino acids. Next is your growth factors, certain proteins, hormones, lipids, and minerals. So these are the things which is basically required for the serum containing media, required for the proper growth and development. Next is serum free media, where serum is not added. It supports the culture of single cell type and incorporated definite quantity of purified growth factor, lipoproteins, and other protein. Next is chemically defined media. So these contains basically contamination free, ultra pure inorganic and organic ingredients. So this basically contains ultra pure inorganic and organic ingredients and may also contains pure protein additives. Last one is protein free media, where no protein will be added. Next is tissue transplantation. You know, there are lots of, you might heard about organ transplantation, kidney transplantation, liver transplantation, and many organs nowadays. Skin also can be transplant. So there are different organs of our body can be transplant in case of different organ failure. So transplantation is also known as xenotransplantation. So tissue transplantation is also known as xenotransplantation. So this transplantation refers to the replacement of uh, when there is a replacement of failed human organs, when the human organs by the functional or functionally animal organ. So when there is a failure of the organs in case of humans, so there will be, we can say, functional animal organ can be implanted in case of in that failure cases. So that will basically support the proper growth and development of that of human. So xenotransplantation or heterologous, it is xenotransplantation is also known as heterologous transplantation, because heterologous matlab in case of human we are basically fixing a organ of animal. So that is basically heterologous. No? That's why it is called a heterologous transplantation. So this is basically transplantation of living cells, tissue, and organs from the specific species to one another, such as cells, tissue, organs are called as xenografts or xenotransplants. So these xenografts, the graft according to their use. So this question was asked in Gpat 2022. Recently, this question was asked. Autografts. So what is autografts or autologous grafts? So when autografts are the grafts in which the donor and recipient is the same individual. If in case of same individual, the transplantation is there, so that is basically called as autografts or autologous graft. Next is isograft. So isograft is the graft between the donor and recipient of the same genotype. The graft between donor and recipient of same genotype. So this is basically called as homozygote or isograft. So if the organ transplantation is carried out between identical twins, so we call it as a isograft. Okay, autografts means same individual, if between same individual. So we call it as a autograft. Next one is your allograft or homologous graft. So the next one is called as allograft or homologous graft. So these allografts are those in which the donor is the same species but of a different genotype. So in case of allograft or homologous graft, so donor is same species but of different genotype. Okay, that is called as allografts. So next is xenograft or heterologous grafts. So xenografts are those in which donor is a different species from the other recipient. This is called as xenograft. So example of autograft graft between the same person from one part of the body to other area. For suppose you are transplanting skin or transplanting hair or transplanting bone from one part of to another part, that is basically called as autograft. Next is allograft. So this between the different member of the same species. So that is basically called as allograft. So, like kidney transplantation, in case of if you are transplanting one kidney of human to another one, this is basically comes under allograft, or heart transplantation, lungs transplantation, or liver transplantation. These are basically comes under allografts. Next is isograft. So, isograft basically between the identical twins. So, if two identical twins, so you are the transplantation between two identical twins. So, this basically called as isograft. And last one is xenografts between different species. So if there is a transplantation between a human and animal, human that is basically called as xenografts. So what we have learned till now, so we learned about, so in this video, we basically talked about animal tissue culture. So animal tissue cultures are the culture techniques when we isolate the organs and add culture it on a culture medium. So we call it as a culture medium. So initial culture that is formed, which called as primary cell culture. So primary cell culture having limited lifespans, and this basically due to the shortening of the telomerase and the respective DNA sequence associated protein that caps the end of the chromosome. So these primary cell cultures are two types. One is attachment culture and the second one is suspension culture. So attachment culture basically grow when it attaches to the base of the culture medium. This is example is kidney. Next is suspension culture. This can be grow when they are in the floating stage. Example is your lymphocyte. Next is cell line. So cell lines basically when the primary culture, when subculture, when the primary culture further subculture to form a secondary culture. This is called as secondary culture. It is also called as cell lines. So it refers to the transfer of cell from one culture vessel to another culture vessel. Is called as subculture process. The cell lines also two types, finite cell line and infinite cell line. So what is finite cell line? So finite cell line undergoes senescence after finite number of division. So example is that this is possible due to subculture or passages. So normal mammalian cells generally have a finite number of cell lines. Normal mammalian cells having finite number of cell lines. After finite number of division, they started the senescence process. This type of cell line basically exhibit the property of contact inhibition. So this type of cell line basically exhibit the property of contact inhibition, density limitation, and anchorage dependency major property that is exhibited by this finite cell line. Next is infinite cell line. This is also known as transformed cell line. The characteristics of the finite cell line when changes due to we can say carcinogens or virus or any spontaneous that is conversion of finite cell line into a transport cell line. The characteristics that is exhibited by transport cell line includes genetic abnormalities, chromosomal abnormalities, loss of contact inhibition, anchorage independent growth, reduced growth factor requirement, biochemical change, increased glucose transport, and increased lactate transport. Next is there are few example of cell lines that is basically used for the biomedical research. One is BHK, that is Syrian baby hamster kidney, CHO Chinese hamster ovary, L is human cervical cancer, AL is human fibroblast or mouse connective tissue. This is the first cell line that is basically discovered. Next is MRC5, human lungs, human embryonic lung. Viro is African green monkey, which is human amnion. MPC11 is human myeloma. Okay, next we talked about what the growth phase. So the cell cycle basically exhibit a typical growth phase. So this growth phase having three stages: lag phase, log phase, and exponential phase. So lag phase is the fast phase when the cells basically exposed to a culture medium and the cells started acquainting with the culture medium. So in the states, the duration of the culture medium basically, so duration of the lag phase basically depend upon type of cells and what is the status of their metabolic state during the culture medium. So this period basically, so in this phase, the cell basically recovered from the stationization, reconstruct their cytoskeleton, secrete matrix to add the attachment. Next is lag phase. Lag phase is the phase where cell started continuously doubling. So animal cell normally exhibit the doubling time between 5 to 25 hours. So the length of log phase basically depend upon the seeding density. The seeding density, growth rate of the cell, and density at which cell proliferated. Next is about plateau phase. So plateau phase is also known as stationary phase where growth and death is equivalent. So this phase is basically due to accumulation of toxic metabolite or inhibitory metabolite and as well as the depletion of nutrients. Next, we talked about culture media. So culture media are the media where the cells are basically grow. So these culture media are consisting of amino acids, vitamins, inorganic salt, glucose, serum, source of growth factor, hormones, and attachments. So these are basically two types: human, natural, and artificial. So natural is basically poor reproducibility, and this is basically due. In case of natural, biological fluids are basically used. Next is artificial media. So artificial media basically contains artificially added substances. So the basic component of a culture media includes physical amino acid, vitamins, glucose, balance salt solution, serum for the proper growth and development. Physical property of the culture medium has to maintain the pH should be around 7 to 7.4. Carbon dioxide concentration will be around 5%. Oxygen should be supplied. Temperature will be around 37 degrees, and osmolarity around human plasma. Okay. Next is about artificial media. There are four different type of artificial media is used: serum containing media, serum free media, chemically defined media, and protein free media. Okay. Next is fetal bovine serum is the most commonly supplement used in animal cell culture. Next, we talked about tissue transplantation. There are also known as xenotransplant. There are different types: are autograft, isograft, allograft, and xenograft. Okay. Now we will discuss some of the MCQ. What till now what we have discussed. So let's discuss some of the MCQ. Okay, so first question is uh, embryonic stem cells are derived from fertilized embryo, unfertilized embryo, sperm, or kidney. So embryonic stem cell, remember the embryonic stem cells, the embryonic stem cells are basically obtained from fertilized embryo. So embryonic stem cells, where it is basically obtained? So it is basically obtained from fertilized embryo. So option A is the correct answer. The product which is commercially produced in animal cell culture is insulin, tissue plasminogen activator, interferon, and hepatitis B. So your right answer is your insulin, tissue plasminogen activator. Example is your streptokinase, altiplase. These are the example of tissue plasminogen activators. So they basically dissolve the clot. So by the dissolve the fibrin clot by activating plasmin. Okay. Next is transgenic animals are produced when GH gene is fused with MT gene, GH, GRF, yeah, FIX. So remember, when GH gene is inserted with MT gene, there is a formation of transgenic animals. Okay. Next question is maximum application of animal cell culture technology today is the production of insulin, interferon, vaccine, or edible vaccine. So the right answer is vaccine. So remember these animal cell culture techniques, the animal cell culture techniques is repeatedly used for the production of different type of vaccines for the different disease condition. Next question. So which of the following statement is not true about the graft? So this question was asked in Gpat 2022. Okay. So the question is isografts are the grafts in which the donor and recipient in the same individual. So which of the following is not true? So you have to find which one is not true. Isografts are the grafts which donor and recipient is the same individual. Autografts are the grafts between the donor and recipient are the same genotype. Allografts are those in which donor is the same species but of different genotype. Xenografts are those in which donor is of different species that have of different recipient. So choose the correct answer from the given option. A and B, D are not correct. A and B are not correct. B and C are not correct. Yes, C and D are not correct. So here you can give your answer in the comment section. Okay. So in the comment section, you put your answer. We will see whether you are giving correct answer or not. Okay. Um, these isographs example is the in here the correct answer is option B. Option A and B are incorrect. So isographs are the graft in which donor and recipient is the same individual. So isographs are the graft where donor and recipient is the same individual. Autografts are the grafts between donor and recipient of the same genotype. Next is allografts are those in which donor one of the same species for the different genotype. Next is xenograft are those in which donor is of different species from that recipient. Okay. Next question. So next question is there is a two statement. You have to find which statement is correct. So first statement is cell from the kidney tissue cannot survive independently. Cell from the kidney tissue. Here the question is cell from the kidney tissue cannot survive independently. Second statement is it requires the surface of attachment and survival. Identify the correct statement. Here the statement is statement, both statement are correct, both statement are incorrect. Statement 1 is correct and statement 2 is incorrect. Statement 2 is correct only. So which is the correct answer? Here the option A is correct. Both the statement are correct. So this when the we are talking about kidney, so these are basically talking about primary cell culture. So primary cell culture. So these kidney are, so as you know, primary cell culture are two types. One is one is adherent cell culture. Another one is suspension cell culture. So kidney comes under adherent cell culture. You know what is adherent cell culture? So adherent cell cultures are the type of cell culture. So these are basically grow when it attached to the base of the culture medium. When it is basically attached to the base of the culture medium, or that will basically grow. So that for that, so it requires the surface attachment for the survival. So option A is correct. Next question is name the type of culture which is prepared by inoculating directly from the tissue of a organism to a culture medium. As I told you, this is a species or this is a mouse. So this is a mouse. So from mouse, I take liver, and from liver, I took hepatocytes, or from liver, we culture hepatocyte. We do hepatocyte culture. So this is basically called what? Primary cell culture, secondary cell culture, cell line, या transformed cell culture. So this is basically called as primary cell culture. This is called as what? Primary cell culture. Okay, this is an example of primary cell culture. Next question. Next question is about name the organism which is first cell line was discovered. So first time which cell line is from which species this cell line basically observed. E. coli, sheep, mouse, या Drosophila. So as I know, the first cell line is your L. So L is nothing but mouse fibroblast. So the species is mouse. So first cell line observed is mouse fibroblast or AL cell line, which is derived from the mouse connective tissue. So this basically derived from where? Mouse connective tissue. So this is this mouse tissue connective when exposed to chemical carcinogen. Okay. Next question. Which of the following is a not a part of growth medium of a animal culture? So in case of animal cell culture, which is not a part of the medium? Starch, serum, carbon source, या inorganic source. So you know, so serum is used for the growth. Carbon source is this is a carbon source is your glucose. So glucose or sucrose is for carbon source, and inorganic salt like sodium, potassium, calcium, magnesium can be used. Okay. So the explanation is so growth medium of So the growth medium of an animal cell contains serum up to 5 to 20% serum. Animal growth cell contains 5 to 20% serum. Nitrogen sources, inorganic salts, carbon sources, growth factors, buffer in water and vitamins. So these are basically the composition of the cell lines, composition of composition of a typical growth culture medium. Next question is, name the cell lines of human embryonic log. So human embryonic MRC5 is basically example of human embryonic lung. So this is the example of human embryonic lungs. Next is L is mouse fibroblast. Which is human amnion. Human amnion. Next is HeLa. HeLa is human cervical cancer. Human cervical cancer. So these are the few cell lines that is used for the different biomedical research. So this is all about animal cell culture techniques. So we discussed animal cell culture techniques and some of the important MCQ related to this. So let's give an overview about plant cell culture. So plant cell culture is basically based upon the fact that plant can be separated into their component, like in case of animal. So plant cell can be cultured in a culture medium for the optimum growth and development. So the father of plant tissue culture is Gilbert Herbert Land. So you have to remember Herbert Land is the father of plant tissue culture. So plant tissue culture practice is used for the propagation of a plant under sterile condition for the production of clone. This tissue culture is commonly called as broad term described like a callus culture, cell culture, protoplast culture, anther culture, meristem culture, embryo culture, and organ culture. So the plant cell, plant cell having two major properties which cannot be seen in case of other. Totipotency. So totipotency means plant cell having ability to grow into whole plant. So this a plant cell can ability to grow into a whole plant, to grow into a whole plant. From a plant cell, it can be a grow up into whole plant. Next, plasticity. So if the nutrient medium of the plant adaptation to growth. So, what is the process of plant tissue culture technique? So, selection of a tissue or explant followed by sterilization of this explant. So, first you have to select a plant, tissue or explant, and you have to sterilize that plant. Now, this inoculation of explant into a culture media containing required nutrients. Now, what you have to do is you have to introduce this sterile plant into a culture media. Now, this inoculated culture is transferred to a growth chamber having appropriate physical conditions. Next is plants are regenerated from the next this plant will started regenerating from the culture of explant. Now these plantlets will root and shoot and then transferred into a greenhouse. So this is basically the overview of a plant tissue culture techniques. So you can see from a plant, we select a plant cell. Now this plant cell placed into the culture medium, plant cell basically placed in the culture medium. Now this plant cell undergo multiplication, root formation, shoot formation, then the acclimatization form a complete plant. Next is types and description callus culture. So callus culture is a unorganized mass of cells which form the plant cell multiply. So this is disorganized multiply in disorganized way. The callus cell basically parenchymatous in nature. Okay, the two major hormone that is used for the callus culture is auxin and cytokines. Auxin and cytokines are used as two major hormones. So ideal temperature required is around 22 to 28 degrees centigrade. Next is suspension culture. So suspension culture, meristem culture. So meristem culture is basically used for the virus free plant, and then anther culture, seed culture, protoblast culture are the different type of culture. Plant cell culture are used. Okay, next is tissue culture. Similarly, as you are using the different culture media for the plant tissue culture, animal cells. So similarly, like that only for the animal cell growth, we are also using animal tissue culture media. So animal tissue culture media, this basically contains different meat components such as growth medium, culture conditions such as temperature, pH, and light and humidity. So growth of basically contains nutrient media. First one is inorganic salts. So inorganic basically divided into two subclasses, macronutrient and micronutrient. Macronutrient example is macronutrient means required at higher concentration required, less concentration required. So a higher example is phosphorus, potassium, calcium, carbon, sulfur, sodium, magnesium. Micronutrient is molybdenum, boron, zinc, copper, nickel, manganese, and iron. Next is carbon sources. The plant tissue culture medium are heterotrophs and depend on the external source of carbon. So plant basically depend upon the external source of carbon. Example is sucrose and glucose are source of carbon. Next is organic supplement growth and morphogenesis can be improved by addition of organic component to the tissue such as vitamins like thiamine, which is very essential, nicotine, pyridoxine, amino acid like glycine can be used. Myoinositol, sugar, alcohol are used in the most of the tissue culture media for improving growth and culture media. Next is a gelling agent is required, as you know. So the culture medium should be solidified. A gelling media should be required. Gelling media like your agar is basically used, which is basically a polysaccharide. pH should be around 5.5 to 5.7. Growth in case of plant, it is around 5.5 to 5.7. In case of animal, it is 7 to 7.4. This is basically in case of animal. Then growth regulators. So growth regulators are like these are the plant hormones. Auxin, cytokines, gibberellins, abscisic acid, ethylene. These are basically used. So these are basically description. So ethylene is basically involved in leaf senescence and fruit ripening. Auxin is basically involved in cell division, callus differentiation, inhibition of root growth, reduction of root formation, advantageous root formation. Cytokines, inhibition of leaf senescence, cell division, inhibition of shoot elongation, gibberellin, shoot elongation, release of seeds, somatic embryos, apical bloods, and these are the varieties of function. These are the varieties of function exerted by these plant hormones. Next is about what are the type of media. So there are different type of media used like White's media, MS media, that is mosaic and scoop originating from the media for organogenesis and regeneration, B5 media, N6 media, and niche media. So these are basically niche media for anther culture. So these are the different culture media for growth of plant and plant animals. Okay, that is an overview of both plant and animal cell culture medium. Okay, I hope everyone understand what we have discussed in the today's lecture. So if you have any doubt regarding this two topics, so you can put your doubt in the comment section, so we will try to resolve your answer. Okay, so this is the last slide. Let me explain. So this slide is basically about how this culture technique is takes place. So basically this culture addition of macronutrient solution in the desired amount to a distilled water. Excuse me. Addition of macronutrient solution to desired amount to a distilled water and stir it well. Next, addition of other components such as hormones, hormones sucrose and agar powders, excuse me, uh, such as hormones, sucrose and agar powders, add vitamins, oxygen and other for the autoclave to achieve the better result. Then addition of more water to achieve the final volume of the medium. Then the pH of the medium should be around 5.7 by adding, adjusting the pH by adding zero point 0.1 NaOH or HCl. Next, in case of solid medium, add sugar in the medium and heat the medium for the dissolving the agar property. Next, disperse this medium vessels to the and plug the vessels. Then autoclave at 121 degree for under 15 psi pressure. Then the medium cool down to the room temperature for the growth of the plant cells. Okay, that's all about animals tissue culture and plant tissue culture. Okay, thank you.